IMAR from the University of the Azores and IMAR-UAc University from the UK are leading work package 4 on restoration of the deep sea. The deep sea is the large ecosystem on Earth and it harbors great biodiversity. However, human pressure have increased in the last decade, creating pressures on these vulnerable marine ecosystems. Recent studies have demonstrated that conservation is better achieved with the protection of intact habitats with the restoration activities on the same ecosystems. However, restoration have never been tried in the deep sea. The main objective of Work Package 4 is to apply the lessons learned from terrestrial and coastal ecosystems to develop principles of restoration in the deep sea. We will develop tools and methodologies that will both develop passive restoration activities as well as new tools for active restoration in the deep sea ecosystems. MERCIS will develop case studies in several deep sea ecosystems: canyons, mid-ocean ridges, continental shelf, seamounts, and also in the fjords in the North Europe. Work Package 4 will also promote the integration of deep sea restoration into policy objectives.
One of the study cases within the MERCIS project is the development of tools and techniques for the restoration of cold water corals that have been impacted by human activities. So why study cold water corals? Cold water corals are cousins of tropical corals, but they live in very deep and dark waters below 200 meters. Can form very large communities called coral gardens, and because these corals can grow quite tall and have a very arborescent structure, that they can give habitat to a number of associated species such as crabs and sea stars, anemones, and even fish with commercial value. But because these corals are very slow growing and they can live for many years, some of them can live up to 4,000 years, and they reproduce very late in life, they can also take very long to recover from any disturbance. Therefore, within the MERCIS project, we are developing these tools to help them recover faster from anthropogenic impacts. One of these restoration techniques consists of transplantation of corals from areas that have been impacted. This technique has been successfully used in coral reefs, but within MERCIS it will be the first time that will be used for restoration of deep sea areas.
The deep sea coral restoration experiments will be done in these aquariums at our deep sea lab. Once they come into the lab, we will fragment the colonies into nubbins, and afterwards these nubbins will be attached to some structures that will be introduced back into their natural ecosystem. After some time, the structures with the nubbins will be revisited to assess the survival rate, growth rate, and physiological state, and also to evaluate the restoration of the associated fauna, and therefore contribute to the evaluation of the potential of this technique as a restoration of the deep sea ecosystems.
Corals are very sensitive organisms. First of all, we have to evaluate if they are surviving, the growth rate, the condition of their tissues. Then we can look inside the tissues and evaluate how physiologically they are changing. And for doing that, we use cellular biomarkers that are indicators of their antioxidant and stress conditions during these restoration processes, which is a very important tool that can give us several indicators of how these sensitive corals are able to not only to survive but also to recover and conquer another habitat and build a new community.